Hello, Assalamualaikum warahmatullahi wabarakatuh, and a very good day to everyone. My name is Anisha Fatma, and I'm a marine science second year student in University Malaysia Terengganu. So today, I'm going to talk about the diversity of marine phototrophs. There are 7 subtopics that I'm going to talk about. The first one is the traditional understanding of primary producers that people used to have until mid-1970s. The second one, recognition of PICOCYANOBACTERIA dominance, discovery of UBIQUITOUS PHOTOHETEROTROPHS, and for the timeline of 1990s until 2011, there are 3 significant findings, which are oxygenic photosynthesis using far-red light, discovery of eukaryotic phytoplankton, and the strange phototrophic microorganisms. And the last subtopic is the diversity of light energy transformation systems and the reconsideration of photosynthesis. What is traditional understanding? It is the old understanding that people used to have back then until mid-1970s. So the first traditional understanding that people used to have is that phototrophs are organisms that transform solar irradiation into chemical bond energy in order to grow. Second one is that diatoms and flagellates are the major contributor in primary production in marine environment. Well, it's simply speculated from 2 situations. The first one is because diatoms and flagellates are widely distributed in wide range of water quality, including eutrophic and oligotrophic environments. Second one is because the phytoplankton sampling and detection method using plankton net with mesh size of at least 3 micrometer. So this is a plankton net, or at least the basic things that a plankton net should have. So back before, the mesh size in nets used for phytoplankton research range from 100 micrometer, 70 micrometer, and 60 micrometer. The usage of at least 60 micrometer mesh size in nets for phytoplankton research, resulting in catchment of only phytoplankton with cell or colony cell of more than 60 micrometer used in research. But recently, in the latest phytoplankton research using DNA detection and flow cytometry, it is found that predominant phytoplankton in marine environment is much smaller than those collected by plankton nets. For example, haptophytes, including Phototrophs, is one of major constituent of primary producer. They have a cell size of less than 50 micrometer, which is way smaller than the mesh size used in phytoplankton research, right? So they can easily pass through the net. That is why, until mid-1970s, only diatoms and flagellates are considered as the major contributor in primary production. So the scientists decided to employ plankton net with smaller mesh size around 10 micrometer. However, the dominant phytoplankton in marine environments has much smaller cell size than the cells that can be caught by those smaller mesh-sized ones. The scientists then found that the dominant phytoplankton in marine environments has way smaller cell size than the cells that can be caught by those smaller mesh-sized nets. In late 1970s, using the techniques of fluorescent microscopy and electron microscopy observation, they found a unicellular PICOCYANOBACTERIA named Synechococcus. This Synechococcus is predominantly distributed in the marine environments, especially tropical and subtropical regions. It is called as PICOCYANOBACTERIA because of its small size. Its length is range only between 0.5 to 2.0 micrometer, and its diameter ranging between 0.5 to 1.0 micrometer. And this Synechococcus play a significant role as primary producer in those environments. And then around late 1980s, using high-performance liquid chromatography to analyze pigment and flow cytometry as a cell detection technique, they found another PICOCYANOBACTERIA, but with genus Prochlorococcus instead of Synechococcus. So what's the difference between Prochlorococcus and the Synechococcus? This Prochlorococcus contains divinal chlorophyll A, which is slightly different than the usual chlorophyll A is contained in the Synechococcus. This divinal chlorophyll A is also efficient in absorbing blue light in ocean for oxygenic photosynthesis. This Prochlorococcus is coccoid to ellipsoidal unicellular cyanobacteria with 0.4 to 0.6 micrometer in diameter and 0.5 to 0.8 micrometer in length. Prochlorococcus do not have phycobilly pigments. Phycobilly pigments is the typical light harvesting antenna in common Synechococcus. This Prochlorococcus is also a dominant phycophytoplankton in tropical and subtropical regions between latitude 30 degrees north and 30 degrees south. Next is the discovery of ubiquitous PHOTOHETEROTROPHS. Photoheterotrophs are organisms that depend on light energy to generate ATP through photophosphorylation. The photoheterotrophs do not rely on carbon dioxide as their carbon source. What's differentiated between photoheterotrophs and photoautotrophs is that photoautotrophs makes its own food. So in early 2000s, there are two significant findings. The first one is the wide distribution of aerobic and oxygenic photosynthetic bacteria, or can known as AAPB, with photosynthetic pigment bacteriochlorophyll A. The second is wide distribution of proteorhodopsin containing bacteria. So, what is AAPB? AAPB is actually derived from the non-sulfur purple bacteria named anaerobic and oxygenic photosynthetic bacteria, or also can be known as NAPB. Both AAPB and NAPB have bacteriochlorophyll A as their photosynthetic pigment, but the thing that differentiates between these two APBs is that AAPB have only bacteriochlorophyll A as the only photosynthetic pigment, but NAPB have several species of photosynthetic pigment such as bacteriochlorophyll A, B, C, D, E, and F. AAPB also require molecular oxygen for their growth and bacteriochlorophyll A synthesis, while NAPB need light as their major energy source during anaerobic photoheterotrophic growth, and the organic compounds are the substrates oxidized to produce reducing power and the intermediates of biosynthetic anabolism. In other words, the things that differ between AAPB and NAPB is that an AAPB needs oxygen, while for NAPB can still functioning well even with limited oxygen supply. So, what is proteorhodopsin? Proteorhodopsin is rhodopsin-like protein detected in marine proteobacteria. Rhodopsin is well known as a photoreceptor in vertebrates as a light sensor. Bacteriorhodopsin of halobacteria and archaea can transform light energy to synthesize ATP. Large amount and wide variety of rhodopsin-like genes were detected in seawater by metagenomic analysis. The total amount of proteorhodopsin containing bacteria is equivalent to 13 to 80% of the total amount of bacteria and archaea in marine environments. So, as for these two significant findings, why are they deemed as significant? Because both bacteria are wide and significantly distributed everywhere, and they play a significant role in light transformation in marine environment. So, oxygenic photosynthesis using infrared light. What is chlorophyll A? Chlorophyll A is the main photosynthetic pigment that can be easily found in green plants. It was first revealed in photosynthesis research using land plants and green algae as model organisms. Chlorophyll A is essential for photosynthesis in cyanobacteria, eukaryotes, and prochlorophytes. It plays an essential role in light harvesting, energy transfer, and charge separation in primary reaction in photosynthesis. It also helped to estimate the quantification of amount of oxygenic phototrophs and the activity of oxygenic photosynthesis. The in situ light absorption for chlorophyll A range between 380 to 710 nanometer, and it is defined as photosynthetically active radiation. Photosynthetically active radiation, or PAR, is the range of spectrum that can be absorbed for photosynthesis process, and it is used to evaluate the light strength for activity of oxygenic photosynthesis. Chlorophyll A absorbs most energy from wavelength of violet blue and orange-red light. It usually reflects green-yellow light, and thus most of the organisms containing chlorophyll A will appear green or yellow to our eyes. In 1996, it was reported that a cyanobacterium named Acariochloris marina performs oxygenic photosynthesis using chlorophyll D. So, this chlorophyll D is actually redshifted chlorophyll, which can absorb infrared light for almost 30 nanometer longer in wavelength than chlorophyll A. It aids the cyanobacterium to perform oxygenic photosynthesis using infrared light from 700 to 740 nanometer, which is not effective for common autotrophs, which means that chlorophyll D can absorb near-infrared light which have longer wavelength. So, it is actually useful for organisms that live in an environment that has limited access of sunlight. This Acariochloris species was widely distributed in coastal areas as epiphytes on seaweed. Recently, in 2010, a new type of chlorophyll named chlorophyll F was found in a cyanobacterium isolated from the microbial mat on stromatolite on the coast of Australia. So, what is stromatolite? As you can see in the picture, stromatolite is a microbial reef created by cyanobacteria. It is formed from the sediment trapping and binding and precipitation activity of microbial communities. So this chlorophyll F has maximum absorption at around 720 nanometer, and it can absorb far-red light up to around 760 nanometer in vivo. As redshifted chlorophylls, chlorophyll D and F, utilize far-red light with longer wavelengths. It helps a lot for organisms in an environment with limited access of visible light to undergo photosynthesis, as the absorption of these redshifted chlorophylls are out from the range of photosynthetically active radiation, PIR. Thus, reconsideration of the PIR concept and primary production in marine environments is required. Next, discovery of picoeukaryotic phytoplankton. In early 2000s, only some dozen species in Chlorophyta, Heterocontophyta, and Haptophyta were known as eukaryotic phytoplankton. In 2010, using the techniques of metagenome and flow cytometry in marine and phycophytoplankton analysis, it was shown that a significant amount of picohaptophytes were widely distributed in marine environment. By pigment composition analysis of marine waters, a carotenoid 19-hexanoyl-fucoxanthin, or known as 19HF, was detected in a phycoplankton fraction at area of 40° to 60° north and south. Since PICOCYANOBACTERIA, which is a predominant phycophytoplankton in tropical and subtropical marine environment, do not contain 19HF, and 19HF is a specific pigment that can be found in haptophytes, so estimation was made that the amount of phycophytoplankton belonging to Haptophyta accounted for 50% of the total amount of the phycophytoplankton fraction. Similar estimation was also made that phycohaptophytes formed around 5% of the global phycophytoplankton biomass. In 2007, it was reported that there is an existence of microalgae in an unknown phylum. Through metagenomic analysis of a plankton fraction of less than 3 micrometer in size, helped to discover phytoplankton that did not belong to any known phyla of algae, tentatively named Phycobilephytes. The DNA sequence of these Phycobilephytes is close to Cryptophysian lineage. It is distributed widely in coastal areas of Europe and North Atlantic. The cell number reaches out to 1.6% of the total eukaryotic phytoplankton. Next, in 2011, an uncultured plastic-bearing eukaryotic lineage, Rhapomonads, was discovered with metagenomic analysis. It is close to Haptophyte, but did not belong to any known phyla phylogenetically. This Rhapomonads is widely distributed in coastal areas of Europe and the Atlantic and Pacific Ocean. Since algae in both candidate phyla have not been isolated and cultivated, the characteristics are not yet known. It is possible to find a further new phylum of phytoplankton which has not yet been discovered. The accumulation of knowledge in diversity of phytoplanktons, including unknown ones, might contribute to better understanding of primary production in marine environment. This strange phototrophic microorganisms is actually a unicellular cyanobacterium-like particle, or also known as UCYNA, or it has also gained its name, which is Atelocyanobacterium thalassum. This UCYNA was first detected as cyanobacterium with a nitrogenous gene for nitrogen fixation in pelagic waters. It was detected by metagenomic analysis of nitrogen-fixing bacteria in marine waters. The wide distribution of UCYNA contributes to the nitrogen cycle in pelagic waters. What's strange about this UCYNA is that it lacks photosystem 2, rubisco, and tricarboxylic acid cycle for respiration. So, in light-dependent reaction, there are two types of photosystem, and photosystem 2 is the photosystem that excite ion and split water. The mechanisms of light absorption by photosystem 1 in the particle is still unclear, but it must be contributing to the transformation of light energy into chemical energy. Its lacking of essential gene suggests that UCYNA might form a symbiosis interaction with other organisms. So, there is a possibility that UCYNA might still use light energy for their activity. Phototrophs is the organisms that use light energy as their main source to grow. Phototrophs are also photosynthetic organisms that can perform photosynthesis. Next is photosynthesis. Photosynthesis is a reaction of reducing carbon dioxide into organic compounds like sugar, using sunlight as an energy source. Primary producers are organisms that synthesize organic compounds using inorganic carbon as their carbon source. Primary producers are photosynthetic organisms like land plants, algae, photosynthetic bacteria, and chemoautotrophs. However, recent discoveries of new marine microbes which utilize light energy suggest that the understanding of these terms might not be suitable for the understanding of energy and the carbon flow in marine environments. Based on my previous explanation, we can come to conclusion that these terms actually have way complex definition. Phototrophs are organisms which can synthesize ATP by ATP synthase using the transmembrane electrochemical potential of photon by pumping up protons from the membranous one side to another side based on light energy. And lastly, primary producers are organisms that can produce organic compounds from inorganic carbon and produce ATP using sunlight energy. So, in conclusion, originally, energy flow in the marine environment based on the food chain, starting with oxygenic photosynthesis with carbon fixation. However, recent studies have revealed that the energy flow through the microbial loop is very important, since it is possible that a large amount of bacteria can transform light energy into chemical energy. So the study of energy flow is very important for us to truly understand why and how some organisms can impact greatly. And that is all from me. Thank you.